Okay, so the first part of this chapter is a little bit of a review of what I talked about before at the very beginning of chapter 13 with meiosis, and how meiosis and the making of egg and sperm cells is related to genetics and figuring out what kinds of kids that you can have. So Mendel discovered the basic principles of progeny by breeding garden peas in carefully planned experiments. He used peas, which happened to end up to be quite an advantage. And so why using peas are an advantage is that, first of all, there were lots of different varieties with different features or characters. So I used the example of flower color and the examples I used with you in the previous notes. And that's one example. That he also noticed that certain pea plants grew taller, some stayed shorter, some had the peas themselves were yellow, some were green, some were round, some were wrinkled. So he had all these different traits that he wondered how those traits are passed on from parent to offspring, and hence did a bunch of breeding experiments with these peas. So that's thing one about the peas. The second is that it's easy to control the mating of pea plants. So you can take the pollen, which has the sperm of one plant, and make it fertilize the egg of another plant. So you can dictate the parent, the offspring, what traits those have, and figure out then what the kids will have, and do some genetic studies that way. So it's much easier. It's easy to do that with plants. Also with plants such as peas, they can be self-fertilized, meaning that a flower produces pollen that contains the sperm, but in the center of the flower, in this carpel, they produce eggs. So what you can do with one pea plant to reproduce this one pea plant, you don't need another pea plant. You can take the pollen from that pea plant and fertilize the eggs of that same pea plant, and then get the peas and grow the peas up and get the babies of these pea plants. So that's one thing that's unique about the flowering plants. And then also he could choose to cross-pollinate. So he could self-pollinate with one pea plant, or cross-pollinate and say, okay, I want to take the pollen from this purple pea plant and put it on the carpel of this white pea plant, and therefore cross a purple and a white pea plant.
What is the pistil of the plant? Would that be like?
That's part of the carpel. Yes, yes.
I remember being pistils.
Yeah. All right, and so so looking at this, this next one is that he chose characters that are only either or, meaning that we'll see later on that not all traits are as easy to look at how they're inherited as purple versus white flowers. So we can get blending of genes, and there's some different variations. So he happened to look at traits that there's either or. So they had purple flowers or white flowers, and there was no other choice. Or grew tall or short, or had yellow peas or green peas. And he used varieties that were true breeding. Another way to say true breeding is homozygous. And it says plants that produce offspring of the same variety when they self-pollinate. Homozygous, what does this mean? Same genes, right? And so a homozygous individual would be like big R, big R. So, or they could be little R, little r. Okay, those are two examples of homozygous. And so what this means is that it says plants that produce offspring of the same variety when they self-pollinate. So if you have a plant that's big R, and so let's say this is the big R, let's just stick with the purple and white flower color trait. And the so this one would be purple, this one would be white. If these guys self-pollinated, that means that when they make their egg and sperm cells, they can only make them with a big R. And so therefore, a purple flower self-pollinating itself can only produce purple flower plants, all right, if it's true breeding, if it's big R, all right. This pea plant that's big R, if it self-pollinates, never would be able to make a little r plant.
I just have a question. Like, can you breed like different species?
Yeah, absolutely you can.
So, like,
Sometimes it doesn't work. We'll talk in a later chapter about that. A lot of times it has to do with chromosome numbers. It's more successful in plants than in animals, actually. And we've actually gotten a lot of new species of plants by humans.
like lighter.
All right, and so on. But yes. All right, and so that's right.
Is that how like people like apple pears or like mix different fruits?
Yes. All right, so in a typical experiment, Mendel mated two contrasting true breeding varieties, which is called, this process is called hybridization. So notice here it says mated two contrasting true breeding varieties. So contrasting means that they have different traits, and so or different versions of the trait that you're looking at. So for flower color, he crossed a purple plant with a white plant. So that's the contrasting part here. True breeding means that the purple is bigger, and the white is little r. And so what we found is that with plants is that when you look at a purple plant though, and you got a purple plant, is this the only genotype that a purple plant could have? No, a purple plant could also be what? Big R, little r. Can you tell by looking at them? No. So this is the beauty of the pea plant here and being able to self-pollinate. Because what he did is he took a purple plant and said, okay, if it's big R, then when I self-pollinate and get babies from this plant, I'm never going to get a white plant if this is big R. Because an individual that's big R can never pass on a white gene to their kids. So they're never going to have any white plants. So that's what he did. He took a purple plant and he self-pollinated it. So pollinated it, got the little babies and the seeds, planted the seeds grow up, you get the kids, none of them were purple, none of them were white. He did that over and over, so we got hundreds and hundreds and hundreds of kids from this purple plant. Not one of those hundreds were white, so you can pretty sure that that plant is big R and not big R little r. So that's how we knew that. The white is always going to be true breeding because recessive always has to have two recessive genes. Okay, if it didn't, it wouldn't be white. All right, and so therefore, when these two were crossed and got the kids, this process called hybridization. What this big R means is that in the two chromosomes of this pea plant, they have a big R on them, and these two chromosomes that have this trait have a little r on them. So what happens when this purple plant and this white plant have baby plants here, this purple plant is going to always pass on a chromosome in their egg or sperm cell with a big R. This one is always going to pass on a chromosome with a little r. So all the kids would be big R, little r. Could these two parents have any other kid, any other genotype, with focusing on just the flower color? No, they cannot have. It's impossible for them to have anything but a big R, little r kid. So they were all purple in this case here, and he called this the P generation, and this is the F1 generation. This is hybridization. So the true breeding parents are the P generation. The hybrid offspring are the F1 generation. So this is the hybrid offspring. Then when you cross the hybrid offspring, what generation do we get? F2, right? And so when you cross, it says you can self-pollinate the F2 or you can cross two of the F2. They're the same thing. So when you self-pollinate a purple plant that's big R, little r, it's really the same as crossing two big R, little r plants. And so here we have the chromosome with the big R, the little r. We cross it with another individual from the F1. And so there's a big R and a little r. And then their kids are going to be part of the F2 generation. So how do we figure out what kinds of kids that these guys can have? So when we look at the types of kids that these guys can have, this parent is always going to either pass on the big R or the little r. Let's review why that is, because remember at the beginning of meiosis, here's your two pieces of DNA that are going to make up this chromosome. They're heterozygous. So in interphase, this piece of DNA that has the big R is replicated. So here's the replication, and we get a replicated chromosome made out of two sister chromatids. This little R is replicated. So here you have your two chromosomes that are replicated. Meiosis 1 separates those chromosomes, so you're going to get the big R chromosome here and the little r here. And then the sister chromatids separate in meiosis 2, and you get your 4 cells. These two, this chromatid's going to go to this cell. This side here is going to go to this cell. So these two are going to be gametes that give the kid the big R gene. These two would be the little r gene. So when we say, and look at an individual that's big R, little r, and we say there's a 50% chance they're going to pass on a big R to their kid and 50% chance they're going to pass on the little r to their kid, that's because in meiosis, when they go through meiosis, they make half their sperm cells or egg cells with a big R, half with a little r. And so when we look at our combinations, this parent could pass on the big R, this parent could pass on the big R, and you get a big R kid. Or this parent could pass on the big R, and this parent could pass on the little r, and you get the big R, little r kid. Or you could do this parent with the little r, this parent with the big R. So notice that there's 2 ways that you can inherit this heterozygous nature. And then lastly, you have little r, kind of like foiling in math a little bit. All right. So in this case here, there are 4 combinations. Every single time these parents have kids, these are the 4 combinations that they can have. And so this one is going to be purple or white. Purple, white. So then here we get that 3-to-1 ratio of purple to white. Does this mean that if these 2 parents had 4 kids, one of them is going to be like this, one of them is going to be like this, one of them, no. What this tells you is it helps you to predict the outcome of their kids and tells you what the chances are of the probability of having a particular type of kid. So if I were to ask you for these 2 parents, what's the chance that they're going to have a white child, you would tell me 1 out of 4 chance, which is 25%. All right, so there's 4 possible combinations here, and this one's only 1 of those. So 1 out of 4 possible combinations gives you 25%. So that means that every single time these guys have a kid, they have a 25% chance of having a white kid, or a 75% chance of having, or 3 out of 4, of having a purple kid. So let me relate this now to a Punnett square, which you may remember from 9th grade biology. All right, so Punnett square, let's just review this. What you do is you put the possible gene combinations of the gametes of parent number 1 along the top. I'm going to say parent number 1 and not mom or dad, because pea plants don't have moms and dads really. I'm just going to call them parent is parent number 2. Okay, guys. The first parent, that's big R, little r here, can pass on the big R or the little r. All right, so their egg or sperm cells are either going to have this or this. And this parent is identical, so they can have this or this. All right? And so what you do with the Punnett square is the same thing. You get the same results here with the Punnett square. So let's say this is the sperm and these are the eggs. So this sperm fertilizes this egg, and you get a baby pea plant that's big R, little r. This sperm fertilizes this egg, you get a baby pea plant with a big, did I say big R, little r here? Big R, big R. This one's a big R, little r. This one here, this one fertilizes this one, you get another big R, little r. Notice here, when I wrote up here, I wrote little r, big R. The reason why I was doing that is to show you that there's 2 different ways that you can get this genotype depending upon which parent you got the big R and which parent you got the little r. Normally, we don't really write little r before big R. So when somebody is heterozygous, we put the dominant first, okay? I just did that to show you the 2 different ways that it's inherited. And then lastly, little r, little r. And so this gives you the same data as this with a nice, neat Punnett square. You guys remember those from 9th grade? All right. And so, So that's that. So these are the F2 generation. So when Mendel crossed contrasting true breeding white and purple flower pea plants, all of the F1 hybrids were purple. We just talked about here. When he crossed the F1 hybrids, many of the F2 plants had purple flowers and some had white. So most had purple, some had white. And he noticed that it happened in a 3-to-1 ratio. So a ratio of 3-to-1 purple to white flowers in the F2 generation. And so this picture here just shows you what I was just drawing on the board, the P and the F2, F1 and the F2. So a reason that only purple flowers were affecting the flower color in the F1. So what he meant was that, you know, the F1 hybrids, their big R, little r, they have a purple and a white gene. Only the purple was affecting the color of that, and that's why they appeared purple. So he called that gene dominant and the white one recessive as a result of that. And he did a lot of genetic experiments, and so he looked at 6 other characters or traits as well, not just flower color, and he found the same results, that when he did the P, F1, F2 generation, in the F2 generation, he always got this 3-to-1 ratio when he did it in all of his different traits. And so at the time when Mendel was doing this, he didn't know about genes and chromosomes. He didn't know all of that we know now. And so he called it a heritable factor, but that we now call it a gene. So the word gene hadn't been coined yet. And so this picture here shows you his actual data and some of the traits that he looked at in pea plants. So you can see flower color, we've talked about that, flower position, seed color, seed shape, a couple different traits about the pods, the height of the plants. So notice here it shows you the dominant and the recessive trait in each one of these crosses. So purple cross with white. Then they gave, this is his actual data from his F2 generation. So he did the P generation F1 and got his F2 generation data. Notice he had 705 purple pea plants to 224 white pea plants. So what he did to get this ratio here is reduced it by taking the smallest number, which is 224, and dividing it into both of these numbers. So 224 goes into 224 once, 224 goes into 705, 3.15. So we got 3.15 to 1 ratio for his purple to white flowers. And so then notice he did this with all of his these crosses, and look at his data here. These are his actual ratios, but when he reduced the ratios, they're all right around that 3-to-1. It was really important for him to have big numbers. So notice here, like especially with the seed color, like 6,022 that he counted that had yellow plants. He had a lot of data. Why is that important?
More reliable.
Accuracy. So the more data you have, the more accurate your data becomes. And so you get closer to what you would expect, all right? So like for instance, if you flip a coin and you have technically a 50% chance of getting heads or tails, but you can flip twice and get both heads. But if you flip 10,000 times, the statistic in your data become closer to what you would expect in a 50-50 ratio. All right, same kind of thing here. And so we got a 3-to-1 ratio for all of his data. So he developed a hypothesis to explain this. And there are 4 different concepts that make up his model about explaining this 3-to-1 ratio. And so and we relate that now to genes and chromosomes. He didn't know anything about genes and chromosomes. So the first concept is that you had alternative versions of genes. So purple versus white. Remember, he had no idea about anything about genes, chromosomes, or anything. So he came up with that concept that there are different versions of your trait. What do we call those different versions of our traits? Alleles. All right. So we have 2 different trait versions: purple and white flowers and flower color. Those versions we call alleles. So this particular trait has 2 alleles, and every pea plant has some combination of those 2 alleles. All right. And then each gene resides at a specific locus. Locus is the same as location on a specific chromosome. And so remember that here's a homologous pair of chromosomes. that it's made out of DNA, that DNA, so here's your DNA, DNA is made out of genes that code for proteins. So in this section of the DNA, there's a gene that has the right order of bases to code for a protein to determine flower color. So this is the purple gene. Then in the homologous chromosome, why this is homologous is because the DNA is similar and that the same genes are in the same spot. So in that same spot on this chromosome, there's a gene to determine flower color. It doesn't have to be the same version of the gene, but it's a flower color trait. And so a flower color gene. So this one has a white flower color gene, this is a purple flower color gene on these two homologous chromosomes. And so this pea plant is heterozygous, having big R and little r as a result of that. So what I'd like you to do next is from this picture to the next picture, I'm going to have you guys fill in the blanks because you should be able to fill in the blanks based on what.
I have a question. That single chromosome, does it represent the other sister chromosome as well, or just like a single one after?
This is before replication. So it's not technically, it's confusing in our way that our book does this because it says it's technically before it replicates, it's really in a chromatin format, it's not a chromosome. Yeah, so when this DNA replicates before cell division, it would look like this, and right here would also be a purple flower color gene. All right, does that make sense? All right. So looking at the second concept is that for each character, an organism inherits two alleles, one from each usually parent. Mendel made this deduction without knowing about chromosomes. The two alleles at a locus on a chromosome may be identical or the same as in a true breeding plant of Mendel's P generation. Just remember, true breeding means homozygous, which means they're either big R, or little r, but the alleles are the same no matter what. So then, alternatively, the two alleles at a locus may differ or be different, as in the F1 hybrids. F1 hybrids are always heterozygous, big R and little r. The third concept is that if the two alleles at a locus differ, then one, which is the dominant allele, determines the organism's appearance, and the other, the recessive allele, has no noticeable effect, because if the individual is big R, little r, the alleles are differ, only the dominant one shows up, right, and therefore affects the phenotype. In the flower color example, the F1 plants had purple flowers because the allele for that trait is dominant. The fourth concept, now known as the law of segregation, states that the two alleles for a heritable character separate or segregate during gamete formation and end up in different gametes. And so that's what's going on here. In meiosis, the two alleles, the big R and the little r, segregate in meiosis 1 or separate into two different cells, which then makes that you only have one of every type of chromosome in your gametes. Thus, an egg or sperm gets only one of the two alleles that are present in the somatic cells of an organism. This segregation of alleles corresponds to the distribution of homologous chromosomes to different gametes in meiosis. So in the process of meiosis, the different alleles segregate and end up in different gametes at the end. And this accounts for the characteristic 3-to-1 ratio he observed in this F2 generation. We can figure out possibilities by using a Punnett square, a diagram for predicting the results of genetic cross. The Punnett square shows you the possible gametes because that's what you put along the top and along the side is the possible gametes. Okay. And capital or uppercase letter represents the dominant allele, lowercase letter represents the recessive allele. All right, and so this picture here just shows you what I was talking about with up on the board here. So this is the Punnett square, is the same Punnett square that I drew up here. They use P's though. I'm not a fan of P's. It's hard to distinguish between upper and lowercase P's, I think. All right, so all right. So then the next 5 bullets or so, we're gonna play this game again and maybe fill in the blanks. You like this game?
like a wide highlighter.
So let's go with this. An organism with two identical alleles for a character you should have is homozygous. So therefore, if they have two different alleles, they're heterozygous. Unlike homozygotes, heterozygotes are not true breeding because they have two different alleles. For the next one, therefore, we just because of the different effects of dominant recessive alleles in organisms, traits do not always reveal its genotype. Yeah, genotype would work. I put genetic composition. Genotype is that is what genotype is. So if you write genotype, that would be absolutely correct. And what that means is that if you look at a pea plant and it appears purple, do you know for sure if it's big R, little r? No. All right, so that's what that means. And so it doesn't always reveal its genetic composition. And so therefore we distinguish between phenotype and genotype because just because two organisms have the same phenotype doesn't necessarily mean that they have the same genotype. So an example of flower color in pea plants, the pictures use big P, and big P, little p. If you used ours, that's fine. So they have the same phenotype but different genotypes. And so I want to show you a couple of things with this picture here. So this is the F2 generation. Here are your four possible combinations. Here's your 3-to-1 ratio in the phenotype ratio. So notice on the bottom it says 3-to-1 is your ratio for that. And then along the right-hand side, it's showing you that these three purple plants, while they appear purple, don't have the same genotype. So we have different genotypes for that. And we can get a genotype ratio of 1 to 2 to 1. So 1 homozygous dominant, 2 heterozygous, and 1 homozygous recessive. In general, the general rule for writing a ratio, whether or not it's a phenotype or genotype ratio, is this. So if we're looking at a phenotype ratio, and if you're asked to do a genetic cross and figure out the outcomes of what kinds of kids that they could have and write the genotype and phenotype ratio, this is the format in which you need to do so. All right, so what you do is the blank is the number. So however many individuals show the dominant trait, and then you put a colon, and then you put a line here, and however many individuals show the recessive. And you could write the actual, so this phenotype ratio 3-to-1. So this is, they're assuming that you know dominant goes first and then recessive, so 3 purple to 1 white, all right, because that's the general rule for writing a phenotype ratio. For a genotype ratio, the format is always you write the number that have the 2 dominant genes, so I'll use ours here, colon, and then the number, whatever that is, that has our heterozygous, colon, and the number that are homozygous recessive, or little r. And that's the format for your genotype ratio. And so in this here, when looking at this at a 1 to 2 to 1 ratio, knowing that format, we know it's 1 big R to 2 big R little r to 1 a little r. So I'm going to give you, let's do a little practice problem here. What I'm going to have you do is, we're going to do a Punnett square. We're going to cross a purple pea plant with a white pea plant. So the first thing is that you have to figure out to do a cross is you have to know the genotypes of the parents, right? So these are the parents. Purple is parent number 1, let's say. White is parent number 2. What do we know about the purple plant? To be purple, it has to have at least one what? It has to have one big R, right? It has to have a purple gene in it, right? Am I giving you any information with this about whether or not that purple is big R, or big R, little r? I haven't. And so I'm going to give you a piece of information here. Let's say that the purple pea plants parents are, let's say that the parents of these guys, or this one, is parents were purple, one was purple, and one was white. And so these guys, these two parents had this purple plant. Does that help you?
Well, it depends if the purple plant is.
Originally, you know that there can be 3 choices.
From this information, you should be able to tell without a doubt
Yeah.
what the purple plant is. No
It's
question.
big or literal.
How many people think it's big R, little r? How many people think it's big R, big R? There's only 2 possibilities. A lot of people aren't sure. Think about the parents. What would the genotypes of the parents be?
Saying that they're the degenerates.
Yeah, what these two parents here to have this purple plant. Think about this purple plant gets 1 gene from mom, or one parent number 1, 1 gene from parent number 2.
Hetero.
Oh, because the white has to be.
White,
Oh my
the
god.
white parent is little r, right?
Yeah.
Without a doubt, the white then has to give the little r to this. This white parent is only going to give a white gene. So therefore, it doesn't matter what this other purple parent is. You know for sure that they had to give that to the purple gene, but you don't know. It doesn't matter what the other gene is. Does that make sense?
Yes.
Okay. All right. So what would the white be? Little r, little r. So therefore, what I'd like you to do is a Punnett square, and then what I'd like you to do is write the genotype and phenotype ratio of the outcome of the kids, the possible kids that they could have. Yeah, just kind of off to the side there.
What would the ratio be if it was like, if they were all dominant? Would you just write 4?
Yeah, no, because you have to write 4 purple or 4 white, because if you just wrote 4, you wouldn't know which one it was. Unless you wrote 4 to 0. Then you would be able to write that, because you would know the 4 goes with the dominant. Yeah, you can write 0. Okay. So in setting this up here, this purple pea plant could give this big R or a little r. The white could give a little r or a little r. So you only have 2 kinds of possibilities of kids just focusing on the flower color trait for this. So therefore, the phenotype ratio in this here, these 2 would be 2 purple to 2 white. You could write like here, they did 3 to 1. You could write 2 to 2, because knowing that how we write the ratio, that it's dominant first and then recessive, so you know it's 2 to 2. The genotype ratio, you don't have any, so you have 0 big R. You have 2 big R little r's and 2 little r's. So you could write it like this. You could also write it like this. Why would it be important to include the 0? Why couldn't I just write 2 to 2?
Is that not things that you talked about, like dominance, like just to show that there's 0% chance to get homozygous dominance?
That's right. That's right. We wouldn't know which genotype these 2's go with. All right, and so you have to include that. So Shane asked earlier. I don't know if people heard. Like, what if you got like a cross where all of them were purple? So like in a phenotype ratio, all 4 were purple. Do you just write 4? No, because then you don't know, is it 4 the recessive trait white, or is it 4 purple, and so on. So, but if you wrote 4 to 0, then we would know that it was 4 dominant. before recess. All right, so we have to be clear.
Can you leave them as 2 to 2 or can you reduce it?
You can, that's a good question. You can reduce it. Is a 2-to-2 ratio the same as a 1-to-1?
Yeah.
Yes.
But you need that for the second one too, even though there's.
Yeah, yeah, you can reduce them. Yep, absolutely. All right. So what I'm going to have us do is there's one other concept I want to do, and that's because there's a few problems on the problem sheet that you picked up here that's going to be your homework that are about this. And so we're going to fast forward a little bit in our notes. I'm going to have you look in your notes and flip, keep on flipping until you see the coin tossing page. It's coin tossing pictures on the top. Go to the next page. Coin tossing picture, that's the easiest way to tell you. So keep on flipping until you see the coin toss, and then at the very next page. All right, very next page. The next page has this picture on the bottom. We're going to look at the set of bullets right above that.
Wait, are we just not?
No, we're going to come back. I just that there's a few problems that have to do with this concept here that I want you to know about. We're going to go back and do everything else once you So what we've talked about so far with purple and white is what we call complete dominance. When you have an individual that's big R, little r, the big R gene is completely dominant over the white, and so therefore we only see purple and we don't see white. And so there's a phenomenon called codominance that I'm going to hit more on later. It says 2 dominant alleles affect the phenotype in separate, distinguishable ways. So sometimes you can have 2 dominant genes and they both show up. But this is the third bullet is what I want to get at and talk about today is this concept called incomplete dominance, where we have the F1 hybrids. So those are the heterozygous individuals. One gene is not dominant over the other, and they both end up making proteins and they blend, and so you get a mixture or a phenotype or physical appearance that is in between both of the parents. Let me give you an example. An example of that. And this flower color in snapdragons is an example. So here we have in the P generation, red crossed with a white flower. All right, the P generation is homozygous or true breeding. So the red plant has 2 red genes. Now notice the way our book writes the red genes. They do a capital C with a superscript with R. The C means that it's the color gene, all right, for the color of the plant. The superscript R means it has a red gene. So therefore, the red plant has 2 red genes. The white plant has 2 white genes. So when they mate and they breed, the red plant always gives a red gene. The white plant gives a white gene. So all of the hybrids in the F1 generation are heterozygous. They have a red and a white gene. But unlike with purple and white flower color in pea plants, this flower color in snapdragons, one gene isn't dominant over the other. So therefore, this pea plant makes the red protein and the white protein. They blend together, and you get a phenotype or physical appearance that is blending or in between the red and the white plant. And so therefore, all of these kids in the F1 generation appear pink. This is why they don't use big R's and little r's, because big R and little r makes the assumption that one gene is dominant over the other, and that's not the case in this particular example. And so therefore then, to get the F2 generation, you cross 2 of the F1s. So a pink plant is either going to give the red gene or the white gene, and you cross it with another pink that can give the red gene or the white gene. And so they can have a kid that gets the red gene from both parents, and they're going to appear red. They can get a red and a white gene, which is going to make them appear pink. They can get a white and a red gene, which will make them appear pink, and a white gene to appear white. And so there are some traits that this is the case. And so what this does is change the phenotypes. So we're still crossing 2 heterozygotes, kind of like we did here, except these 2 here appear the same as this because these guys, the big R was completely dominant over the little r. Now when you have heterozygotes, in this example here, you get a blending and you get pink. So if you're looking at a trait that is inherited this way, and you look at a snapdragon but, and look at the color of its flowers, do you know for sure what its genotype is? Yes, you do. Because if it's red, you know it has to be 2 red genes. If it's white, it has to have 2 white genes. And then pink, it has to have a red and a white. That's different than over here in complete dominance. When you have complete dominance and the purple was dominant over the white, looking at a purple flower, you don't know if they're big R, big R, or big R, little r. Here, the heterozygotes look different. They blend, all right? And so, so that's where I want to leave off today. And so your homework is this genetic problems here that I want you to do on a separate sheet of paper. Some of the problems, there's a few of them that the trait is incomplete dominant, so they show, you'll see red crossed with white and so on and so forth. So there are some problems like that. Okay. What was that?
So we have to go on a separate.
No, I don't know.